Welcome to our journey into the fascinating world of zoology. Today, we are going to explore how biologists organize the incredible variety of animals on Earth, specifically focusing on a group called chordates. Whether you realize it or not, you are a chordate, and understanding this classification system is like reading the family tree of our own evolutionary history. We will start from the very basics of how we group animals and work our way up to using real mathematical tools to measure the health of ecosystems. Let us dive in. To understand how we organize life, imagine walking into a massive supermarket to find a specific cereal. You do not wander randomly. You find the grocery section, then the breakfast aisle, and finally the right shelf. This is exactly why biologists use taxonomy. With millions of animal species, we need a reliable system to organize them. Chordate classification is our supermarket map for a very specific group of animals, those sharing a core structural blueprint, like a spinal cord. By grouping these animals based on shared features, we can easily see who is related to whom and how they have adapted over millions of years. Now that we understand why we classify animals, let us look at what makes a chordate a chordate. In biological terms, we are categorizing animals into hierarchical groups based on shared evolutionary traits. To belong to the phylum Chordata, an animal must possess four very specific anatomical features at some point in its life cycle. These are a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a postanal tail. Even if an animal loses some of these features as an adult, like humans losing our tails, having them during embryonic development is enough to secure their place in this phylum. Knowing the four key features, we can now divide this massive phylum into smaller, more specific groups. Scientists split chordates into three main subphyla. First, we have Urochordata, which includes marine animals called tunicates. Second is Cephalochordata, representing small fish-like lancelets. The third, and most familiar to us, is Vertebrata, animals with backbones. We then break the Vertebrata subphylum down even further into familiar classes. These include Pisces for fishes, Amphibia for frogs and salamanders, Reptilia for snakes and turtles, Aves for birds, and Mammalia for mammals like us. This structured hierarchy is the foundation of evolutionary biology. Before we look at a visual map of chordates, we need to understand what a phylogenetic tree actually is. Think of it as a grand family tree that shows the evolutionary history and genetic relationships among diverse creatures. Every time a branch splits on this tree, it represents a major evolutionary milestone or a new adaptation. For example, the development of jaws was a massive leap that separated early jawless fishes from more advanced ones. Later, the incredible adaptation of the amniotic egg allowed certain vertebrates to leave the water entirely and conquer land. Understanding these branching points helps us read the history of life. Let us visualize this hierarchy with a phylogenetic tree, which is essentially a family tree for species. On the far left, we start with the ancestral chordate. Notice how the tree branches out. The first splits separate the Urochordata and Cephalochordata from the Vertebrata. As we follow the vertebrate line, we see key evolutionary milestones acting as branching points. The development of jaws separates jawless fishes from cartilaginous and bony fishes. Further along, the evolution of lungs branches off to amphibians, and the incredible adaptation of the amniotic egg gives rise to reptiles, birds, and mammals. This diagram beautifully maps out millions of years of evolutionary history. You might be wondering why we spend so much time meticulously sorting and naming these animals. The primary reason is that classification provides a universal language. It ensures that a researcher in Japan and a researcher in Brazil are talking about the exact same species without any confusion. Furthermore, this system is absolutely crucial for conservation efforts. By mapping the tree, we can identify which unique branches are most at risk of extinction. In medicine, knowing how closely related certain mammals are to humans helps us understand diseases and develop life-saving treatments. Without this system, our understanding of biodiversity would be a chaotic mess. Now that we have a solid grasp on how to classify chordates, we need to apply this knowledge in the field. When biologists study a specific habitat, simply listing the names of the classes present is not enough. We need a reliable, objective way to measure exactly how diverse and healthy that ecosystem is, and to compare it over time. True biodiversity is not just about counting the total number of species, it is also about how evenly the individual animals are distributed among those groups. To capture this balance, scientists turn to mathematics, using specific formulas to quantify biodiversity based on our classification data. This brings us to the Shannon-Wiener Diversity Index, a powerful mathematical tool used by ecologists. Conceptually, this formula calculates the uncertainty or variety in a community. Let us break down the symbols. H prime represents the final diversity index itself. S stands for the total number of different chordate groups we have classified in the area. The variable p sub I is the proportion of individuals belonging to one specific group, found by dividing that group's count by the total population. Finally, ln is the natural logarithm. Because proportions are decimals between 0 and 1, their natural logs are negative. The negative sign at the very front of the formula flips our final result into a positive number. When the number of groups increases, or when populations are more evenly balanced, H prime increases, indicating a healthier ecosystem. Let us see this formula in action with a concrete example. Suppose you survey a small pond and classify 50 fishes, 30 frogs, and 20 turtles. First, we find the total population, N, which is 100 animals. Next, we find the proportion for each class by dividing their count by 100. Fishes are 0.50, frogs are 0.30, and turtles are 0.20. We then plug these proportions into our Shannon diversity formula, multiplying each proportion by its natural logarithm. Adding those negative values together gives us negative 1.03. Finally, the negative sign at the front makes it positive. Our final answer is an index of 1.03. This indicates a moderate, healthy level of diversity, with a fairly even spread across the three classes. Now that we have calculated a baseline for our healthy pond, let us consider what happens when the environment is disturbed. Imagine 5 years pass and severe agricultural pollution affects this exact same pond. When we return, we might find that the total number of animals has not changed, but the types of animals surviving certainly have. Pollution often wipes out sensitive species while allowing hardy species to dominate. This drastically shifts the distribution of individuals. As we're about to see, the Shannon Diversity Index is specifically designed to detect this loss of evenness. It mathematically penalizes ecosystems that lack balance, heavily weighting the decline of rare species. Let us calculate the new diversity for our polluted pond. Our new survey finds 90 fishes, only 5 frogs, and only 5 turtles. The total population N is still exactly 100, but the proportions are now heavily skewed. 0.90 for fishes and 0.05 for both frogs and turtles. We plug these new proportions into the formula. Notice that the natural log of 0.05 is a large negative number, reflecting the heavy mathematical weight placed on rare species. When we multiply and sum these terms, we get negative 0.395. Applying the final negative sign gives us a new index of 0.395. This drastic drop from 1.03 mathematically proves that the true diversity has collapsed due to pollution. We have covered a lot of ground today, so let us review our key takeaways. First, chordate classification is the essential framework we use to organize animals that share traits like a notochord. By grouping them into subphyla and classes, we can trace the grand evolutionary story connecting a fish to a mammal. Second, this system is not just academic, it is a universal language vital for global conservation and medical research. Finally, we learned that by applying mathematical tools like the Shannon Diversity Index, we can move beyond just naming species. We can actually measure the health, complexity, and balance of ecosystems. Mastering this classification system is your first major step toward understanding the interconnected web of vertebrate life on Earth. Great job today!